Chapter 2, Section 6, Cell Surface Structures. Okay, we're going to be looking at here things that are attached to the cell envelope but are actually outside the cell. Okay, first ones we're going to be talking about are what are referred to as capsules and slime layers. Okay, these are going to be polysaccharide coats, many sugars that are outside the cell envelope. They are an outer covering of some sort. If it's this outer covering of sugars, these polysaccharides are tightly packed, really tight, somewhat rigid, it's called a capsule. If they're loosely compacted, loosely attached, and very hydroscopic, meaning they're pulling in lots of water, they refer to that as a slime layer. Yeah, it's a little hard to see here, but this picture from the book, figure 216, this A here. This is a loop, like you're going to play with in lab, that they've touched to a colony on a petri dish. This slime layer is such, all the millions of bacteria that have this slime coating, that it actually is like pulling snot, slime up, where it's stuck to the metal loop. You look here in subfigure B. Here you're seeing everything around the bacteria, that's what these spots are, bacteria, everything around has stained dark to India ink. But these halos you see around the cells, that's actually the capsule. The capsule is creating this protective, as you see here, this protective outer barrier. Things can get through, small ions, small nutrients can get through, water can be exchanged, but many chemicals like antibiotics and stuff can't pass through. Same with this slime layer. Things can't get through. The functions of these capsules, these slime layers, well, it helps in attachment, okay? Bacteria don't want to be free floating around when they can attach to a surface and then let the liquid and all the nutrients in the liquid free float around them as it just flows over them. Also, there's going to be some safety in numbers. So if one can attach, remember, 1 becomes 2, 4, 8, because these things will grow and other things will attach to them as they start to clump. You can get the formation of a biofilm. Many bacteria forming layers, and all these layers have different capsules and slime layers. They start to form protective coating for everybody in lower layers. Capsule slime layers also help with infectivity. It's actually harder to treat with antibiotics microbes that have capsules and slime layers, because like I said on the last slide, antibiotics, it's hard to get those to pass through. Antibiotics can't get into the cell, can't do damage to the cell. Cell's not going to die because of the antibiotics. Many of the cells in your immune system, that their job is to clear out infections. Well, before they can clear it out, before they can kill it, they have to be able to grab hold of it so they can phagocytize it. Well, can't grab a hold of a slime layer. Hard to get up and around a capsule. And these slime layers and capsules, especially the slime layers, because remember I said these are very hydroscopic, they're pulling in all the water they can to remain slime consistency, which means they're going to prevent dehydration, the drying out, and the death of the cell. Other microbes can have alternate things. Some of these are going to be referred to as pili. Pili are short projections off of the surface of the cell. They are protein-based. They are outside the cell, maybe attached to the cell wall, passing through an outer membrane if it's a gram-negative. These pili allow for adherence to surfaces, like your teeth or, anywhere on your body, in your body, that they happen to be found. Allows them to stick together to also start to form biofilms. Pili come in multiple forms. The first one is a shortened pili referred to as fimbria. Fimbria are more for attachment. Whereas some of the other longer pili, well, they can take on jobs such as what is referred to as a sex pili. The sex pili are long projections. You get two bacteria in close proximity that have these long projections, and they will link up. And it's a hollow tube. What allows for the passage of DNA, a copy of the genome, or a copy of a portion of the genome, or a copy of a plasmid, to be passed from a donor to a recipient bacterial cell. They have what they call electrically conductive pili. They do kind of the same thing, but it allows for transfer of electrons, signal molecules, back and forth. And then there's the type 4 pili that allow for pathogens to adhere to our tissues. You know, say like your lungs or your heart valves, things like that. They are hollow, so they'll also allow for the passage of proteins from the bacterial cell to the target cell, things like toxins and inhibitory molecule proteins. The type 4 pili also allow for motility. They can wiggle a little bit, which will aid in movement of the bacteria, especially if they're attached to something and they want to move around on it. That little twitching helps to move them. It's not great, it's not fast, but they're not stuck in place. So these short little projections you see here are a type of pili known as fimbria. These long ones you see here that are much longer than the cell itself, those are another one we're going to talk about here in a few minutes, known as flagella. Here's those sex pili. So this isn't from one cell. This is from two different cells. Somewhere in the middle here is where they met. And this is hollow. So it allows, if this is the donor is either passing a copy of a plasmid over to the receptor or to the receiver, or it's making a partial copy of its chromosome and with a few genes on it, and that's what it's sending over. We also see that there are these things referred to as hamas, or the plural hemi. These are going to be grappling hook-like things that are going to be on archae. These allow for attachment, they allow for movement. They're similar to the type 4 that I just talked about a few slides ago for bacteria. But here, these hemi, they have barbed ends that allow more for attachment and movement. So you'll see them, they extend out, they're big things, but they're going to have these hook-like structures at the end. So they go and they can attach to and hold that archaebacteria in place so that, you know, the fluid movement's not carrying them around. They're held in place and the fluid just moves over them. Any nutrients in the fluid moves over and around them so that they can absorb it that way.